Okay, so we are starting with today's class. I hope I didn't got very late to add you all. I'm very sorry for the delay. So this is Kanika Gupta, your TA for the session, and today we'll be discussing about week 4 lectures. And as our classes generally go with, we'll first revise what we have studied in the last week, and then we'll discuss the question of the assignment. Meanwhile, if you guys have any questions, you can ask me, okay? So, yes, last week we've studied about the genome, the central dogma of the biology. Before going into the detail of the genome and central dogma of the biology, there are some terms that you guys must be familiar with. So, one of these term is hereditary. As you have noticed that there are some kind of a traits which are similar in kids from their parents and between the siblings. So this is just all happens because of heredity. What is heredity? Heredity, also called inheritance, is the passing on of traits from parental generation to their offsprings, either through asexual reproduction or sexual reproduction, and the offspring cells obtain the genetic information of their parents. So basically, the information which is getting passed from parents to their offsprings is what we call is the transfer of that information is called as the heredity. Basically, what a child inherits from their parents in the physical traits, like in the eyes, in the heights, ears, etc. So these are all heredity, which is inherited by a child from its parents. So, what is the history of the genetics? How come we got into this heredity, gene, genome kind of terms? Where from this came? So, for that, it is a well-known truth that the qualities are passed down from one generation to next generation, which is called heredity, and the offspring shares certain characteristics with both of their parents. So, how does this happens? This question was answered by Gregor Mendel, which is also called as the father of the genetics, who is also called as the father of the genetics. He was a mathematician by studies, but he studied this heredity through its mathematical terms also only. What he did is he actually studied it using the sweet peas. He grew the sweet peas in its garden, and using the mathematics, it he studied that there is something which is getting passed on from one generation to next generation, which is telling that generation what your traits will be. That was called as gene later on, but he did it on the sweet pea which he grew in its garden. He counted it mathematically, and he found out that some offsprings resemble to one parent in one trait, and in another trait with another parent. So he did that in the late 19th century by three biologists, Hugo de Vries, Carl Correns, and Eric von Schermark. So basically, when Mendel was there, his work was not much recognized because people thought how a mathematician can tell all of these things, and how can he justify all of this biology things. So they did not pay much attention to his work. So later on in the 19th century, these three scientists, Hugo de Vries, Carl Correns, and Eric von Schermark, actually proved what Mendel said. They worked on Mendel's work and proposed that different characters have individual hereditary carriers, and that inheritance of specific traits in organisms comes in particles. De Vries called them as pangenes. Gene term hasn't been came till now. It was called as the pangenes. Gene term came out later on. Almost 20 years later, Wilhelm Jonasson and William Bettison proposed that term gene. So gene term came nearly 20 years later, when these three scientists proved that there is something which is getting transferred from one generation to next generation. And these two scientists gave that thing name as genes, and the study was called as the genetics. Okay. But Edward Strasberg and others continued to refer to the basic physical and functional unit of heredity as pengene. Sometimes scientists still use the term pengene, but nowadays we use the term gene for to denote this transfer of characters from one generation to next generation, the factor which is responsible to transfer these characters. Okay, so genes are present in chromosomes which are evenly distributed between two daughter cells during cell division. Biochemical studies show that chromosomes are composed of proteins 60% and DNA 40%. Where from these proteins come from, we'll discuss this further in the lecture. So, the whole chromosome is actually a tightly packed DNA, and this DNA is packed around some proteins. So that is why chromosome is made up of 60% of proteins and 40% of the DNA. Until 1940, proteins were thought of a genetic material because proteins are polymers made up of 20 different types of amino acids, which are abundant and encode diverse information, because each of the information which comes in the form of DNA is further conveyed in the form of proteins only. So that is why it was thought that the proteins are the actual genetic material, but it is not the case. It was finally found out that it is the DNA which is the genetic material, which is getting transferred from one generation to next generation. Okay, so the properties of genetic material: for a molecule to be considered as genetic material, it must accomplish certain properties. What are these properties? It should be stable, it should be heritable, it should show some kind of a mutations which offers evolution, actually, which is the one of the many reasons for the evolution. And fourth is the replication, it should be able to replicate. So these four must properties a genetic material should have to be called as the genetic material. Okay, so there are some direct evidences, experimental, and some indirect evidences which says that DNA is the genetic material. Okay, just a moment, class. To join. Yes. So yes, as I was telling, there are some evidence, some direct, some indirect evidences which tell us that DNA is the genetic material. In this slide, there are three experiments that we will be looking through, which prove that DNA is the genetic material, and it is the DNA, or we can say the nucleic acid, which is transferred from one generation to the next generation. So in 1928, it was the Griffiths experiment. In 1944, it was the Avery, McLeod, and McCarthy experiment, and then the Hershey and Chase experiment, which again proved that it is the nucleic acid which acts as a genetic material. So in Griffith's experiment, what he did is Griffith identified two strains of Streptococcus pneumoniae, a bacteria. So he identified two strains of the Streptococcus pneumoniae bacteria, the virulent and pathogenic strain known as the S strain and smooth strain or S3 strain, and the avirulent strain was known as the R strain. So the strain which will cause, which is pathogenic, is virulent strain, and it is called as the S strain. The strain which will not be pathogenic is called as the avirulent strain, and it will be called as the R strain. Okay, so the S strain has smooth outer coat made up of the polysaccharides, and R strain जो है they lack this polysaccharide coat. That's why their surface appears rough. That is why they are called as the R strain. So what they did is, as I told you, R strain is the avirulent strain, S strain is the virulent strain. Okay, so what they did is, they did four case studies. In one case, what they did is they provided the mice with this mouse injected with the S3 strain. Okay, S3 strain bacteria, and the mouse died. Obviously it will die because S strain is the virulent strain, it is pathogenic, it produces toxins. Then in case 2, the mice was given R2 strain, in which the mouse survived because it is a virulent strain. Then what happened is, in case 3, the heat killed S3 strain was given to the mice. And heat kill strain means the, because he high temperature can disrupt the binding between the DNA double strands, so that is why this heat killed strain uh, didn't show any effect on mice. So the mice did not die. In the next case, what they did is they gave the bacteria R2 strain, the live strain, and heat killed S3 strain. Now the mice died. Why it died? Because when this heat kill S3 bacteria was given there, the DNA got incorporated into the R2 strain. R2 strain took up that DNA and it repaired that DNA, it used that DNA, and then the R2 strain become the virulent and it killed the bacteria. Okay, so it showed that there was something in this S3 strain which got transferred to R2 strain, and which led to the death of the bacteria. And from here, it was found out that it was a nucleic acid which was, there was some genetic material which was transferred. Okay, next was the every McLeod and McCarthy experiment. Guys, just a moment, I need to keep my charger. Okay, so next was the every McLeod and McCarthy experiment. In this, what they did is, uh, before their experiment, it was believed that genetic material was made up of proteins, as I told you already. So they work on transforming principle in Griffith's experiment to investigate its biological makeup, biochemical makeup of it. So to determine which biochemical from the heat killed S cells could have transferred to our cells, and so that our cells become virulent, they isolated proteins, DNA and RNA separately from the S cells, and finally found out that DNA from S bacteria was all that was needed to convert our bacteria. How they did that? uh, they actually provided enzymes for each of these things. Like to one mice they gave heat killed S3 bacteria and live R2 bacteria and proteases. What does proteases do? They degrade the proteins, they digest the proteins. So when it was given, mice died. So it showed that the S2 colony was, when the culture, when the cells from the gut of this mice was taken and grown on the culture, it was showing that S3 colony was living again. Why S3 colony? Because now the R2 has taken up this DNA, and now they are growing as the virulent strain. Next experiment, what they did is they heat killed the S3. Again, they gave the live R strain bacteria, and now they treated this with RNAs. RNAs are those enzymes which degrade the RNA. After which mice died again, which showed that the RNA is also not the thing which is getting transferred, which is making the bacteria virulent. Then in the third experiment, they treated it with the DNAs, and in that case, mice survived. So from there, they proposed that the genetic material which is getting transferred from one generation to next is DNA, not RNA or protein. So this is how they explained that. So in conclusion, they found that both RNA and proteins digesting enzymes had no effect on transformation, proving that molecule undergoing transformation was neither a protein nor an RNA, it was a DNA. So DNA is the genetic material. After that, Hershey and Chase did another experiment. They research on virus that infects E. coli and provide additional evidence for the genetic importance of the DNA. So the DNA core of the T2 bacteriophage is encased in a protein coat. I'll show you. I'll just check if no, I don't have the picture, but it is actually the we can say the capsid or capsule kind of a part of a virus, the proteinaceous part, and in which the genetic material is stored या फिर it stays. ठीक है? So they put this theory to test in the following manner. They radioactively labeled the T2 phage in either their proteins or DNA component before injecting them into the bacteria. So radio labeling means the heavier isotope of that particular element was used. In case of sulfur it was 35 sulfur, and in case of phosphorus it was 32 phosphorus was used. So as you all know, phosphorus is the component of the nucleic acids, the phosphate group, and sulfur is used in the proteins in the amino acid like cysteine, methionine, etc. Okay, so they use the heavy isotope. Specifically, they infected known radioactive Escherichia coli with radioactively labeled T2 bacteriophage, and after doing that they injected the bacteria with T2 phages that had been radioactively labeled either in the DNA component or in their protein component. Okay, so the infected bacteria was agitated in blender and two fractions were separated by the centrifugation. One fraction contained the empty phase coats. That means the see, whenever a phage infects the bacteria cell, it leaves the genetic material inside the bacteria cells, and the caspid, or we can say the outer coat, remains outside the cell. So when they did the centrifugation, one fraction contained the empty phase coats that were released from the surface of the bacteria. These consisted of the proteins and therefore carried the 35 as a radioactive label. Because it doesn't enters inside the cell, so they're left outside, and the genetic material entered inside the cell of the bacteria. So the radioactivity due to phosphorus was detected from the bacteria themselves. So it showed that the genetic material was DNA only. Okay, so this was what Hershey and Chase experiment was. Then after, there are some indirect evidences also which says that DNA is a genetic material. DNA is regularly present in nuclei of all types of cells. Equal amount of DNA is present in all cells of an organism. Amount of DNA is proportional to the ploidy of the cell. That means either it is having single copy of that DNA or two copies of that DNA. They are telling about that. So the amount of DNA is proportional to the ploidy of the cell. Sorry, haploid cells have half the amount of DNA than diploid cells. Okay, so nuclear divisions occurs only after DNA duplication during S phase of the interphase. अगर DNA genetic material नहीं होता, so there was no requirement of the S phase, it would have divided as such only. But no, DNA is important, and which happens, the replication of the DNA happens at the S phase. Okay, फिर different species have different amounts of the diploid DNA. Out of all macromolecules, DNA is metabolically most stable. Okay, indefinite number of combinations are possible with four bases A, T, G, C. Okay, and DNA has some physical and chemical properties in all organisms, yet allowed to produce great diversity of organism. Okay, DNA has that physical and chemical properties uh, due to which it can be diverse in different organism, like mutation or hereditary is also one of the thing, because different DNA are coming from different parents, which is causing the variation or diversity in the DNA of different animals. Okay, now if RNA is a genetic material, we have to prove that RNA is not always the genetic material. So for that, according to the RNA world hypothesis, RNA was the first genetic material that stored all genetic information, and it is believed that the first life arose from it. RNA is thought to catalyze a number of chemical reactions in primitive cells. The presence of two hydroxyl groups in ribose sugar increases their activity. Basically, RNA is very unstable, because ek to one, it is single stranded, another it has two OH group available at its 3 prime position and 4 prime position, due to which sorry, at its 2 prime position and 3 prime position, due to which it makes it increases the reactivity of RNA with the other molecules present inside the body of the organism. So that makes it very unstable, and that is why it can't be called as the genetic material in all cells, because these properties of RNA make it very unstable. It is not structurally stable. So yes, the genetic material should be stable. We have read about it already. Ultimately, these unstable molecules are replaced by more stable genetic molecules. During this stage of evolution, DNA molecules emerged, and they have replaced RNA's role as both genetic material and structural components. The unstable and degraded nature of RNA has led to the development of double stranded DNA genetic material that is both chemically and structurally more stable. Okay, however, RNA is not completely eliminated. They still serve as genetic material in some systems like viruses, and they catalyze few essential biochemical reactions in the cell. Also, the complex machinery of protein synthesis from DNA is still proceeding through RNA. Okay. Uh, Next is there are some viruses, because as I told you, RNA is not the genetic material in all the cases, but it is genetic material in some cases. So the virus consists of two parts, nucleic acid and protein coat. Okay, sometimes with additional envelope. Protein coat is also present with sometimes an additional envelope. So viruses contain only one type of nucleic acid, that is either DNA or RNA. Viruses having RNA are called as the riboviruses. They vary in structure of their nucleic acid. Most of the plant viruses have RNA either single stranded or double stranded. The RNA can be single stranded or double stranded in different viruses in which they are present as genetic material. So the variation can occur at that part also. Uh, the first evidence in favor of RNA as a genetic material came from the tobacco mosaic virus, in which RNA is present as the genetic material. So it was the Frankel and Conant and Singer demonstrated in 1957 that progeny viruses from TMV infection with viruses having RNA from one strain and protein from another were invariably of the kind determined by the RNA, not the protein. So they did the experiment and proved that it is RNA, which is the genetic material in case of the TMV. So, in conclusion, what happened is Frankel and Conard therefore came to the conclusion that both DNA and RNA carry genetic information. As a result of all of these investigation, his research established that genetic material for TMV is stored in RNA rather than protein. So protein is still not the genetic material. It is either RNA or DNA. Mostly it is DNA, but in some cases, in some viruses, it is RNA also, and that can also be single stranded or double stranded. That varies in different viruses. But RNA serves as a genetic material in few instances. Okay, now we have understood what is the genetic material and what is the molecule that we call as DNA and how it is carrying the information. Now we should understand the term genome. What is genome? So genome is actually the complete set of DNA comprising of nuclear and mitochondrial DNA in any organism is its genome. The total genetic part of that organism is called as the genome. A total hereditary material present in any organism. It may be either DNA or RNA. Genome is a totality of chromosome unique to particular organism or any cell within the organism. Each genome contains all of the information needed to build and maintain that organism. So genome has all the information which is required for that organism. Okay, how genome and genotype differs? The total hereditary material is a genome. The information contained within the chromosome, small portions of the genome is the genotype. Okay, so the total hereditary material is genome and the information contained within the chromosome. A small portion of the genome is the genotype. Genotype means genotype is actually the expression is actually we are saying the types of gene for a particular trait. Okay, you will understand this better when we'll discuss further the phenotype and genotype term distinctly. So, what types of genome are present in different kind of organism? In prokaryotes, it can be double-stranded DNA. It can be circular, it can be one chromosome, or it can be a nucleoid. Any of these can be present as a genetic material in case of prokaryotes. In eukaryotes, it is mostly double stranded DNA which is linear, which makes many chromosomes and which is stored inside the nucleus, not lying freely in the cytoplasm as is the case with the prokaryotes. In case of organelles genome, the heredity may represent in mitochondria and the chloroplast. So basically, mitochondria and chloroplast also do have their own genome. So it is called as the organelle genome. Then the viral genome can be DNA, can be RNA, can be single stranded, can be double stranded, can be circular, can be linear, depending upon different viruses. So viral genome is of much diversity in this case. Then this genome must be organized in the organism, so that this it can be organized in a way that whenever the information is required from DNA, it can be accessed easily, and also the genetic material stay prevented from all of different kind of attacks happening inside the cell or on the cell. So genetic material must be organized so that it can be stored properly. So in a cell, each DNA molecule associated with proteins, and each DNA molecule and its associated protein is called as the chromosome. As I told you, in any chromosome, 60% part is the protein, 40% part is the DNA. So they are talking about that protein only. Organization holds true for prokaryotes and eukaryotes both. In both of these organisms, the organization or packaging of the DNA into chromosome occurs the same way. So the packaging of DNA in the chromosomes serves several important functions. Chromosome is compact form of DNA that readily fits inside the cell. It protects the DNA from damage. Only packaged DNA can be transmitted efficiently to both daughter cells when a cell is dividing. Because if the DNA will be present haphazardly, not arranged properly in the form of chromosomes, then it would be very difficult to distribute the chromosome equal and um almost similar to both the offspring, so that there can't be any kind of abnormalities. So it should be arranged properly for that. So why the packaging of DNA is required? The DNA compaction is required, uh, such as in the nucleosome and higher order chromatin fibers. The physical side of DNA is to reduce the size of the physical size of the DNA significantly, so that the transfer of DNA can be easier. Okay, next is the DNA protection. The densely packed chromatin structure shields DNA from exposure to potentially harmful enzymes and radiations, and it also helps to prevent DNA from becoming tangled or breaking during cellular processes. Regulation of gene expression, because not in every organ every gene is required. Some are required and some you needs to be suppressed, so that there won't be crowding and only the required proteins can be synthesized. So yes, it helps in the gene regulation. It helps in the replication and repair, that we all know, because replication is a process in which the whole DNA must be doubled. So it should be arranged properly so that the DNA can do the replication part in a systematic manner. It won't interfere in the DNA replication. Okay. It also ensures accurate chromosome segregation that happens in case of the mitosis or meiosis. Mitosis we have already studied. So when the genetic material or chromosomes are arranged at the metaphasic plate, then it shows that it has been arranged properly, and now equal amount of genetic material can be distributed to each of the cells. Okay, why the packaging of DNA is required? Because it enables the regulatory interactions. The regulatory interactions uh, must be managed, so that not excessive protein and not less protein will be formed. It will form exactly the amount of protein which is required by the body. In case of prokaryotes, how the organization happens? They have small bodies and small genomes, so they do not contain a well defined nucleus, so it must be compacted in a way that it won't hinder the growth of the bacteria, and also whenever the genes are required it is accessible to those genes. Okay, so they have a single chromosome that floats in the cytoplasm. The genome size ranges in between 10 raised to the power to 10 raised to the power 6 base pairs with a high density. Apart from this single chromosome, some bar exist either. Some bacteria have extra chromosomal DNA called plasmids. So there are some extra chromosomal plasmids also, which is actually beneficial for the bacteria because it provides some kind of a resistance to the bacteria, additional nutrients to the bacteria, or some additional benefit to the bacteria. So these are the extra chromosomal DNA. That means it is not the part of the original chromosome, but they are present inside the bacteria. They have come, they have entered the bacteria from outside. Okay, so plasmids are actually what prokaryotes frequently carry one or more smaller independent extra chromosomal DNA called as the plasmids. Plasmids are not genomic DNA, they are accessory DNA molecules, small circular DNA molecules that have the ability to self replicate. Unlike the larger chromosomal DNA, plasmids typically are not essential for bacterial growth. ठीक है, so unlike the larger chromosomal DNA, plasmids typically are not essential for bacterial growth because it is providing some additional benefit. It is not the one which is carrying the genetic information for the cell. It is carrying the genetic information but not that information which is vital for cell to grow or to divide or to live. That is why. So plasmid provide advantages to the bacteria such as antibiotic resistant, herbicide resistance, etc. In addition, unlike chromosomal DNA, plasmids are often present in many complete copies per cell, so that the amount of protein production can be increased. Okay, so the bacterial genome, bacterial chromosome DNA is usually a circular molecule that is a few millions nucleotides in length. Escherichia coli, E. coli 4.6 million base pairs, and Haemophilus influenza 1.8 million base pairs. So typical bacterial chromosome contains a few thousands different genes, and structural genes sequences account for the majority bacterial DNA. And the untranscribed DNA between adjacent gene are termed as the intragenic regions, which is introns in case of the humans, or in case of the larger animals. Okay, so the packaging of DNA, how does the DNA is packaged? So prokaryotic cells typically have smaller genomes. They need to compact their DNA in is still substantial. E. coli must pack its 1 mm chromosome into a cell that is only 1 micromolar in length. It is less clear how prokaryotic DNA is compacted, but it is compacted. It makes a single chromosome, but it does make. Okay. Now the next thing is nucleoid. What are nucleoids? A prokaryotic chromosome is circular and resides in a cell region called as the nucleoid. It is still lying in the in the cytoplasm, but prokaryotic chromosome is circular and resides in a cell region called as the nucleoid. It is also having some kind of a proteins to protect the DNA from any kind of the damage, chromosome from any kind of the damage. Only one complete copy of their chromosome that is packaged in a nucleotide. 80% of DNA by mass and can be unfolded by agent that acts on the RNA protein. The proteins responsible for condensing and maintaining the supercoiled structure of DNA have not been identified yet. DNA determines what proteins and enzymes and organisms can synthesize, and therefore what chemical reaction it is able to carry out. ठीक है, DNA determines what proteins and enzymes an organism can synthesize, because DNA has all the information, it is the genetic material, therefore what chemical reaction it is able to carry out. ठीक है, key features वही है, it should have an origin of replication, it should be stable, it should be what, there was the four features that we have studied for any genetic material to be proven as the genetic material, and these were the four features: the stability, the heritability, the mutation, and the replication, self replication. ठीक है. So this was about the prokaryotic genetic material folding. Now, in case of E. coli chromosome is compact, one fifth of the cell volume. It has to be compact this much so that it can freely move inside the cell volume, as it does not have a defined cell membrane. The determinants of nucleoid folding: negative supercoiling by topoisomerase, condensation by the attachment of nucleoid structure proteins. So, a nucleoid is highly condensed during a rapid growth. RNA polymerase concentrates in transcriptional loci. RNA-P is distributed throughout the chromosomes. ठीक है, RNA polymerase concentrates in transcriptional loci, and RNA polymerase is distributed throughout the chromosome so that it can make the RNA from the chromosome, and then the proteins can be formed. In case of eukaryotes, how does the genome organization happen? So, in case of eukaryotes, genome is linear and conforms the Watson and Crick double helix structural model. Ki basically the base pairing and the helix turn. So, base pairing, we all know it is A double bond T, G double bond triple bond C. So, this is the Watson and Crick double helix structure model says. This is embedded in the nucleosome complex DNA and protein structure that pack together to form chromosomes. Eukaryotic genome have unique features of exon intron organization of protein coding genes, representing coding sequence, and intervening sequence that represents the functionality of RNA part inside the genome. Okay, so eukaryotic genomes ka they have this unique feature of the exon and intron part. Exon is the one which is the coding part, intron is the one which is the non coding part. Okay, a human haploid cell consists of 23 nuclear chromosomes. So it is a set of 23 nuclear chromosome in a haploid cell. If as the most cells of humans are, or any other organism are, they are diploid in nature. So the number of these chromosomes will be 46. That is the diploid cells, and have the double the number of the chromosomes. Okay. Eukaryotic chromosomes are usually linear. Each typical chromosome is tens of millions to hundreds of millions of base pairs in length. Eukaryotic chromosomes occurs in sets. Many species are diploid, which means that somatic cells contain two sets of the chromosomes. Repetitive sequences are commonly found in near centromeric regions. Telomeres contain specialized sequence located at both ends of the linear chromosomes. So these sequences are the actually ones which will help later on in the cell cycle to the fibers that are actually going to pull these chromosomes apart and make the new cells. So these proteins are actually helping in that. Okay, so the chemical composition of chromatin, it contains 20 to 40% DNA, 5 to 10% RNA, proteins is 55 to 60%. Now, what are those proteins which are actually embedded inside the DNA and which gets roll on when the function is complete, and it opens up when the DNA needs to be accessed for further process. ठीक है, for formation of the different proteins and etc. So histones are those proteins upon which this DNA wraps and stays stably inside the nucleus. So it is found in all eukaryotic cells. Commonly present histones in eukaryotic cells are H1, H2A, H2B, H3, and H4. Core histones are two occupies of the core histones from the position core around which DNA is wrapped. It is not the part of the core protein, but it associated with the linker DNA which links two types of the nucleosome. So linker DNA is the one which is actually attaching two nucleosomes, and the core histones are the one which upon which the DNA wraps, and it makes the one nucleosome each. Each octacore will make one nucleosome. Okay. So this is just the molecular weight of different type of the core histones and linker histones. Next is the assembly of nucleosomes involved order association of histones with DNA. So first H3 and H4 will make homotetramer. Okay. Then this H3 and H4 will be associated with the DNA. After that H2A and H2B will come into action, and it will form the complete octamer, and then the nucleosome is formed, and the genetic material is ready to arranged as a chromosome. So nucleosome building blocks of the chromosome. As I told you, nucleosomes are the one which actually get arranged in the coil form or in the solenoid form, and then the then upon these nucleosomes the DNA part is wrapped, and then after wrapping up of the DNA, these nucleosome get attached to the nucleosome in the vicinity through H1 linker, and this is how this H1 histone is useful. Okay, model is a scientific model which explains the organization of DNA and associated proteins in the chromosome. It also further also further explains the exact mechanism of the folding of DNA in the nucleus. The model was prepared by Roger Kornberg in 1974 and is the most accepted model of chromatin organization. So what it says is the nucleosomes will be arranged, and upon these nucleosome the DNA will be bound totally. It will be wrapped around them, and then this nucleosome will arrange to make the whole chromosome. Okay. So this is how it is showing H4, H3 core DNA and H2, HB. So H2, HB, H3, H4, all four of them will replicate and will make the octamer. ठीक है, and this is the stages of the formation of the chromosome. So first of all there is this DNA which is lying freely on the cell surface. Then beads on a string form of chromosome, first order of placing nucleosomes, and then this will be further complex down, and finally we will get the chromosome. The C value paradox is the new thing which again you guys should know what is C value paradox. So however complex is the organism, just a moment guys. So however complex is the organism, more complexer will be its genome, because as much as complex is the organism, it will be needing more information, and that more information will be stored in the larger and complexer genome, so that all kind of information can be transferred from one generation to next generation. So, sorry, that is what C value paradox is. The C value, the quantity of DNA per haploid genome such that seen in the nucleus of a spermatozoon, is used to describe genome size in eukaryotes. Because size is essentially consistent within a species, it is known as the C or constant or characteristics. The mismatch between the C value and the presumed amount of genetic information contained within the genomes was called as the C value paradox. Okay, the mismatch between the C value and the presumed amount of genetic information contained within the genome was called as the C value paradox. Since we cannot assume that a species possesses less DNA than the any required to specify its valid function, we have to explain many species contain their amount of excess DNA. ठीक है, so it is C paradox is basically the complexity of the genome. Okay, next is the unpacking of DNA. Now the DNA has been tightly packed. It is perfectly there in the form of the chromosome, staying in the cell without getting affected by any kind of enzymes or any kind of attack. So there must be something which helps in unpacking of the DNA, so that whichever gene we want, we can access it, and we can transcribe it and then translate it to make the required protein, so that cell can function properly. So for that we have three main covalent modification: the acetylation, the phosphorylation, and the methylation. Now acetylation is an acetic acid group, so it will provide a negative charge to this histones, and the negative charge of histones will repel the negative charge of the DNA, and that is how the DNA will be opened up for our requirement, like whatever kind of gene we want to amplify, it will open up. Same goes for the phosphorylation. It will provide the negative charge to the histones, and the negative charge uh, on the DNA will actually uh, repel this, and that is how the DNA will be opened. In case of methylation, there is no charge imparting, but yes, it causes the crowding and change of the environment there. It actually neutralizes some kind of amount so that the interactions can be weakened between uh, the histones and the DNA molecules. So these three molecules actually help in unpacking of this chromosomes. Now we have some specialized chromosomes in our in nature. That is a polytene chromosome first. Polytene chromosomes, which are gigantic chromosome that grew from smaller yet developing chromosome, frequently appear in the salivary glands of Drosophila flies. ठीक है, ड्रोसोफिला फ्लाई जो होती है हमारी फ्रूट फ्लाई, उसकी सलाइवरी ग्लैंड में बहुत देखा जाता है दिस पॉलिटीन क्रोमोसोम. दे आर नॉन एज द सलाइवरी ग्लैंड क्रोमोसोम्स बिकॉज दे वर फाउंड इन द सलाइवरी ग्लैंड. E.G. Balbiani found polytene chromosome in the salivary gland nuclei of the Chironomus midge larvae in 1881. Due to the presence of several chromonimata in them, they are known as the polytene chromosomes. Most polytene chromosomes are located in the interphase nucleus of a few cells in diphterian fly larva. Each chromosomal component is successively duplicated as they grow from the chromosomes of the diploid nuclei. After each DNA doubling, the later stage of the mitosis are eliminated, leading to the development of the polytene chromosome. ठीक है, so that is how these polytene chromosomes are made. And in this polytene chromosome you will be seeing some kind of a puffs which are being formed in between. So these puffs are actually the active region of these chromosome. They are called as the Balbiani rings because the scientist's name was Balbiani who discovered this. So these are actually the active sites of the chromosome where the transcription is going on. Okay, next is in the line is the lamp brush chromosome. Lamp brush chromosomes are transcriptionally active chromosomes which are mainly found in the germinal vesicles of the large oocytes of many vertebrates and invertebrates. Okay, the lamp brush chromosome derived their name from the lateral loops and extrude from the chromosome at certain points. Okay, they are very much transcriptionally active. If we have picture, you can see they have some kind of a loops formed in them, which loops those loops are actually the active part of this chromosome. Now there are many loops formed on a single chromosome, so it gives an appearance of the lamp brush, so that's why it is called as the lamp brush chromosome. Now the next part is the central dogma of the life, या फिर another terms we call it as the central dogma of the biology. So what happens, what this central dogma is, it is the core reaction which happens all the time so that our body or any other organism can function properly. In this the DNA is in this the process is the RNA will be formed from DNA and protein will be formed from RNA. The DNA can replicate to form the DNA, and RNA also can replicate to form the RNA through RNA polymerases. Okay, so the protein is made up of amino acids, and every protein has unique amino acid arranged in a specific sequence. So the information to synthesize proteins with unique amino acid sequence is provided by nucleic acid present within the nucleus. In a preset reaction sequence, DNA present in the nucleus give rise to a specific RNA sequence, and that in turn guide the cellular machinery to synthesize proteins. Scientists considered this as the fundamental event to run the life and considered as the central dogma of the life. In another word, Francis, in his communication to journal Nature, states that the central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information. It states that such information cannot be transferred back from a protein to either protein or a nucleic acid. But it is not the case. It happens in some cases, but those are the exceptions. Those are not the actual processes happening all the time in all the cells. Okay, so as I told you, three processes: replication, DNA can replicate itself; transcription, the formation of RNA from DNA; and translation, the formation of protein from the RNA. Okay, so the process in which the DNA is formed from RNA is called as the reverse transcription. Same goes, there are many different processes that can occur in nature, but again, those are the ones which are not naturally are all the time occurring processes. It is a chance event or specific to some organisms. Okay, so this was what was read in the last week's lecture. Now we'll come to the assignment questions. Before that, if anyone have any questions, they can ask me. Otherwise, we'll proceed with the assignment questions. Should I proceed with the assignment questions? Okay, I'm proceeding with the one. So the practice goes the same way. Everyone will write the answer to that question in chat box so that everyone can participate. Okay. Okay, guys. So question first: genes are present in following which are evenly distributed between two daughter cells during cell division. Genes are present in where? Answer to this question is yes. The answer to this question is C. They are present in the chromosomes. Okay. Next question. Next question says in a Griffith experiment, transformation was prevented by DNA's digestion, indicating that following is responsible for carrying genetic genetic information. It was Griffith experiment or McCarthy experiment. There's some confusion with the question. Mm, it was McCarthy experiment. It's okay. So in this experiment, the answer to this question is 8 minute guys chat box. Yes, it is again C. It is the DNA, which is the genetic material. Okay. Next question. Following contains all of the information needed to build and maintain that organism. What is that? Again, the answer is C. It is the genome part which contains all the information. Okay. Next question. In prokaryotes, apart from single chromosome, some bacteria have extra chromosomal DNA called. क्या कहते हैं उसको हम? Yes, it is called as the plasmid. The extra chromosomal DNA is plasmid. Next question. Each chromosome contains a centromere that forms a recognition site for the dash protein. What is that? Kinetochore proteins. Yes. Question number 6. The linker histone is which one is the linker histone and which one is the octamer forming histone, the core histone? Tell me the answer to this question. It is D, it is H1. Yes, correct. Mm, next question. The tails are the extensive sites for following, including methylation, acetylation, and phosphorylation. What is that? These three processes are what? What kind of a modifications are they? Post translation modification, correct. The lamb brush chromosomes occur in the dash stage of the prophase of the first mitotic division in primary oocytes of all the animals? Which stage it is? It is the diplotin stage, right? The acetylation takes place at lysine residue K4 and K5 in H4 group through an enzyme called एसिटाइल ग्रुप है, और एसिटाइल ग्रुप यहाँ पे लग रहा है, हट नहीं रहा है, तो इस केस में कौन सा एंजाइम होगा वो? हिस्टोन एसिटाइल ट्रांसफरेज, एग्जैक्टली. It is histone acetyl transferase. Answer is C. Okay. In eukaryotes, DNA is tightly bound to histone proteins, leading to the formation of DNA protein particles called. हाँ जी, बताइए क्वेश्चन नंबर 10 का आंसर. देखें जरा सबने क्या लिखा है. क्वेश्चन नंबर 10 का आंसर इज बी, दैट इज न्यूक्लियोसोम्स. Okay. Next question. In eukaryotes, the lack of a direct relationship between organism complexity and genome size is referred to as what? अभी पढ़ा हमने. What is that? C value paradox. Right. Question number 10. The sequence dependent synthesis of DNA from pre-existing DNA is known as. बताइए क्या है ये? The sequence dependent synthesis of DNA from pre-existing DNA is known as replication. Right, it is the replication, because transcription and translation are different processes. Okay. Next question. In reverse transcription, the enzyme dash synthesizes a complementary DNA strand from an RNA template. It is called as what? Which enzyme is involved? It is B, RNA dependent DNA polymerase, because it is taking RNA as template, so it will be RNA dependent, and it is making DNA, so it will be a RNA dependent DNA polymerase. Primase synthesizes the following at the site of replication to start the replication process. What is that? Primase क्या करता है? Yeah, it will make the RNA primers. Okay, it will make the RNA primer. After DNA unwinds, following binds to the single-stranded DNA to avoid rewinding and helps reduce the energy required for unwinding. What is that enzyme? Yes, it is single strand binding proteins. These are the proteins which actually keep the single strand stable, so that the replication can occur without any kind of the disruption or missing part of the DNA. Okay. So these were the questions that was there in your assignment. If anyone has any doubt in any question, they can ask me, or if anyone has any doubt in the lecture, they can ask me too. I'm here only. You can ask me questions now. This was just a discussion of what we had in the week 4. Yes, everyone. Any questions? Any questions, anyone? You can ask me the questions. In that question, it is not Griffith's experiment. It was every McLeod and McCarthy experiment, there might be some kind of confusion there, but it's okay. The answer will then also be same. When we did the Hershey and Chase experiment, what they do? They use the bacteriophage, not the simple cell. They have used the bacteriophage to study this. That GNA is the genetic material. After that, we studied about the packaging of the DNA, why it is important, and how the packaging happens in case of prokaryotes, how the packaging happens in case of eukaryotes. ठीक है, उसके बाद we studied the central dogma of the biology, that how does all the processes work in a cell, how replication occurs, how translation occurs, how transcription occurs. So all of these processes we have studied about. Apart from that, if anyone has any question, they can ask me. Okay, any questions, anyone? Did you guys understood about the replication, transcription, and translation? So replication is basically, we all know, is the process of DNA की copy होना. ठीक है? DNA forming its own copies is called as the replication. When information is needed to be transferred within the cell, then the DNA transfers the information in RNA. That is, the RNA get copied from the DNA, same as a sequence of the DNA, and then that RNA is the one which is actually helping in formation of the protein, and the information is passing on from one in the different organelles, or in the different between the different signaling molecules inside the cell. So yes, this is how the transcription, translation, and replication occurs. Apart from that, any other questions, anyone? No one wants to ask any questions? No one has questions? Have you guys studied in the last week? Have you guys seen all of the videos? Were you able to understand all of those lectures? Okay, then if you don't have any questions, you can leave the meeting. We can conclude the meeting here only. If you don't have any questions, you can leave the meeting. Thank you so much for joining, everyone. Now you can leave the meeting. Guys, you leave the meeting if you don't have any questions that needs to be answered now. Thank you so much for joining.